Hi everyone, Dr. Wiseman here, and we are doing part 10 of the APHY102 class. This is module 10 on the urinary tract and the fluids involved in the urinary tract. Here are the relevant chapters. Let's get started. Fundamentally, the act of urination is termed micturition, and it involves an interaction between your autonomic nervous system as well as your skeletal nervous system and the bladder and the muscles around the bladder. It is a reflex first and foremost, which is why in very young kids they have not either learned or gained the ability to withhold urine voluntarily. So when the bladder, and it can range anywhere from 200 to 500 milliliters, sometimes more, you get a stretch occurring in the bladder. The bladder is actually called transitional epithelium, which means that it can stretch and retain its function. And what that stretch does is it sends a signal to the brain in the autonomic area to begin contracting to create urination. So it involves both smooth muscle, so it's an autonomic nervous system, as well as skeletal muscle in what we call the somatic or we call the voluntary muscle system. Loss of tone in either of those muscles can lead to aberrations in our normal, what we consider normal adult micturition activity. So the micturition process is actually primarily a reflex. It's a reflex, at least in the first part, and then a voluntary allowance in the latter part. So we begin with enough liquid or pressure in the bladder to send a signal up these, they're called baroreceptors, to the spinal cord in the sacrum. There, what you get is an elicitation of a signal down, parasympathetic. This is all involuntary, and it heads to two places. First of all, it heads to the detrusor muscle of the bladder, where it initiates contraction. So if you think about it, this is actually sort of a positive feedback cycle. You've got pressure that leads to contraction, which leads to more pressure. Okay, could lead to more contraction. Simultaneously, you're going to stimulate relaxation in what's called the internal urethral sphincter. So you've got contraction of the detrusor and relaxation of the internal urethral sphincter. Now, one more thing has to happen, and that is this is skeletal muscle here. This external urethral sphincter must relax, and this is fundamentally skeletal muscle. So if it's not a convenient place for you to micturate and you have the ability, you can hold it for a period of time. All right. The problem is that you're getting a sort of positive feedback cycle, and this can get worse and worse and worse. So you can hold it for a period of time by keeping that external urethral sphincter contracted voluntarily, and then you can release it under appropriate circumstances. And again, if we lose the ability to manage the internal sphincter, then we might have an urge to go more often than we would like. And if you can't keep the external one contracted appropriately, you may be leaking urine accidentally. For anatomical purposes, all of these terms on this slide are relevant. You have the ureters, which are ducts connecting the kidneys with the bladder. Once the filtrate and so forth has got here, it's pretty much going to be urine at that point as opposed to filtrate up in the kidneys. You have the detrusor muscle, which is not well illustrated. It's here, and that's pretty much, there's more than one muscle here, but this one is the most important. All right, so that's the detrusor muscle. Then you have this funny structure right here called the trigony or trigon. This is an area of material that has two urethral openings and also prevents backflow from occurring from the urethra. It's sort of a block to infection, block to misdirection of fluids. Then you have the smooth muscle ring, that's the internal urethral sphincter, And then you have skeletal muscle, which is external urethral sphincter. You have the urethra, which can be of varying lengths. It's shorter in women, longer in men. And lastly, the urethral orifice. With the male anatomy, there are just a couple of variations that we need to point out. Up here in the upper part, with the ureters, the bladder, trigony, and internal urethral sphincter, those are identical to women. After passing through the internal urethral sphincter, you pass through this what's called the prostatic urethra. This is where you receive seminal fluid and fluids associated with reproductive activity. You pass through the external urethral sphincter. Then you have this membranous urethra that was not shown in the previous diagram, but it's there in men and women. And then finally, you have what's called the penile urethra. And the distances are longer for anatomical reasons, but that also renders males a little more resistant to bladder or kidney infections because of the sheer distance that has to travel with a pathogen. Women are much frequently have bladder and kidney infections just because of that sheer distance. Now let's move into the constituents of urine, the product of post-filtration processing in the kidney. Once that filtrate arrives in the bladder, we start calling it urine. One of the most notable characteristics, other than color, shall we say, is odor. And that odor is primarily responsible because of the nitrogenous wastes that are generated from the breakdown of proteins. When we break down proteins into what are called amino acids, one of those groups is an amine. All right. So we have the amine, and then ammonia is just a more hydrogenated amine. It's very closely related structurally to the amine group is ammonia, and ammonia is what you smell, all right? But it's also what we use for certain types of cleaners and so forth. You find ammonia in lots of products that are meant to sterilize things, and that's an important point. Yes, we have a very negative connotation when it comes to urine. But there was a time when urine was actually collected as a tax in Roman times because urine serves as a very wonderful laundry detergent. If you let urine settle, the ammonia will waft away. Ammonia is a volatile, easily evaporates. And so once that ammonia has gone away, you're basically left with a kind of soapy salt water. And that's why it was used as a laundry detergent. And urine is the second cleanest bodily secretion, second only to tears. So it's actually almost always sterile, and in part due to the concentrations of ammonia and nitrogenous compounds, which are natural antibiotics. So we have this urea in here, which is again the product of protein breakdown. It can make the urea molecule proper, or it can also make ammonia, and that's what gives urine its characteristic smell. Just a word of caution, that's what this word means in German, Achtung. The next few slides, I am going to proceed relatively carefully. The main reason being they are a little bit more technical than I think they need to be, so it's important for you to listen to where I'm putting emphasis. I can't just go in and change all the materials. I can't rewrite the book. I can't do a lot of these things. And that's the real value of class in particular is the ability to hear what the instructor is emphasizing and translate that into your notes rather than a pure recitative paradigm where you're just going to write down everything I say and you're not going to prioritize information. We don't have that luxury in healthcare. We're always going to have more information, more knowledge than we can actually keep in the front of our brains. So listen carefully over the next few slides and see where I am putting emphasis. You can then emphasize, and then you can choose to focus in those points of emphasis for exam purposes. So good luck, and again, bring your questions. That's the other thing. One thing that a lot of people don't realize is that our body is literally a massive collection of different compartments. And you can categorize those compartments in different ways. In this case, we have three major, shall we say, fluid compartments that in advanced courses and so forth would be further subdivided. Generally speaking, water can move between these compartments. Not always every single time, but certainly a lot of the time. What happens is we can vary the amount of fluid in one compartment, meaning water content, and then the other compartments will equilibrate to that. So in terms of basic function, we have two major compartments. We have extracellular fluid and we have intracellular fluid. And you can see that intracellular fluid, the fluid inside the cells, dominates this figure. And this is true of the body as a whole, that the intracellular compartment is the majority of mass and water volume in the body. In the extracellular, and there are anatomical compartments, and there are other types of compartments that are extracellular, like for instance cerebrospinal fluid or synovial fluids, et cetera, et cetera. But those are all subdivisions of what we call interstitial fluid, which is the majority of extracellular fluid. That is, of the remaining from the 100%, you can see that roughly 75%, or maybe 2/3, is intracellular. Then the majority of the extracellular fluid is actually not blood plasma, but it is known as interstitial fluid. This can include, like I said, cerebrospinal fluid, synovial fluid, pericardial fluid, the fluid just that flows outside of our blood vessels, but also outside of our cells. Then you have the remaining plasma. So these compartments, we can regulate, for instance, we can regulate fluid volume in plasma by what I mean there is water content. And then the other compartments will shift water to and from based on how we are managing blood plasma. So water will move between these compartments relatively freely. There are exceptions, as always. The water will move, and often we use sodium, especially in the kidney, as a means of affecting that concentration of water in the blood plasma by which then the other ones will equilibrate. So sodium, all right? And that's why sodium is so important in terms of things like if we're going to manage blood pressure, if somebody's hypertensive, one of the first things we'll do is regulate their sodium intake, all right? And kidneys are a big time sodium regulator. Sodium is practically king in the kidneys, I like to say. If we change blood pressure, we change blood volume, like for instance, if we have a hemorrhage or we do something along those lines, the thing that will happen most often, at least for a long-term response, is that the brain and hormone systems will endeavor to tell the kidneys to start retaining sodium in order to retain more water. Here's one of these graphs, which on the surface may seem harder than it actually is. Because we got all this sodium and potassium and calcium, all this good stuff, right? Looks complicated, lots of bars and that sort of thing. Let's simplify this a little bit. First of all, we've got our three compartments. We've got the extracellular compartment, blood plasma, and interstitial fluid. Then we have intracellular fluid. So the two extracellular and the intracellular, the three colors here. Let's start with the blood plasma. Okay, so blood plasma has, and I highlighted it here, a lot of sodium, relatively speaking, a boatload of it. And then it also has chloride. Yeah, it's got a couple of other things. It's got a little bit of protein, it's got a little bit of bicarb. That's nice. That's fine. But the big ones are sodium and chloride. Then let's go to the next extracellular compartment. It also has a lot of sodium and a lot of chloride. All right. And that's true of all extracellular compartments. They tend to be high in sodium and chloride. The only substantial difference between blood plasma and interstitial fluid is that blood plasma has more protein in it. Interstitial fluid doesn't have a lot of protein in it, but it does have a lot of sodium and a lot of chloride. And that's a rule of thumb for extracellular fluid. It's always going to have a lot of sodium and a lot of chloride. It should. Now let's go to intracellular fluid, and what you see right away, a boatload of potassium. So we've been in this talk for about 15 minutes. You have already metabolized several pounds of ATP just to establish this sodium chloride and potassium gradient between the intracellular and extracellular compartments. So intracellular, you're pumping in potassium, and you're pumping out sodium into the extracellular fluid. All right, so we've got lots and lots of potassium. And then we also have this buffer over here. It's phosphate buffer. All right, great. That's it. All right, this looks a lot more complicated. Find the big ones. Sodium and chloride, maybe a little protein for the plasma. Sodium and chloride for the intracellular fluid or the interstitial fluid. And then lastly, potassium and phosphate for the intracellular fluid. And we typically maintain an osmolarity. Sometimes you'll hear the word osmolality. Okay, they are basically synonymous. There's some technical reasons why you would call it one or the other. Think of them as interchangeable. So we're going to keep it, our cells and our tissues somewhere around 290 to 300 milliosmolar, which is sort of like one of those magic numbers like pH of 7.4. Okay. So the big message in that previous slide is that we see elevated levels of sodium and chloride in general in the extracellular fluid. And we see elevated levels of potassium. Phosphate is a little bit of a different story, but potassium is a big one in the intracellular fluid. So let's look at some of these basic functions of sodium and potassium, and then we'll go for one more. It's a little bit of a more complicated story, but it is an important one. Well, sodium is major for regulating extracellular fluid levels. Your kidney controls it through a hormone, often it's called aldosterone. If you've heard of the renin-angiotensin-aldosterone pathway, RAS, we have lots and lots of drugs that go after that in order to help maintain blood pressure and extracellular fluid volume levels. Sodium is also massive in maintaining and establishing voltage across plasma membranes, especially when we're talking about plasma membranes that have a physiologic function of what we call excitability. If you look at muscles, their two major functions are excitability and contractility. Excitability is driven by ion concentrations, and sodium is one of the massive ones for that. So when we have either neurons or muscles, certainly both, when we have variances in the voltage across the plasma membrane, we call that a potential. That's the basis for signaling and initiating responses like muscle contraction, or in the case of a neuron, release of neurotransmitter. And it's also important for when we're not stimulating those tissues, maintaining what we call a resting membrane potential. Sodium is a key component of that resting membrane potential. And we start to deviate in our sodium levels in our bodies. One of the things that begins to happen that's most noticeable are neuromuscular symptoms. And that brings us to potassium, which is actually even more important than sodium when it comes to resting membrane potential. It is the primary driver. It's the one that is most responsible for establishing resting membrane potential. There's a reason potassium chloride is one of the 3 drugs in a lethal cocktail, because if you get deviations in potassium, that can lead to death very, very quickly. All right. It's involved in the repolarization following excitation in action potentials. So when we stimulate a nerve or a muscle, you depolarize first, and then you have to repolarize. And potassium is the ion that's responsible for that. Certainly control in the kidney as well, because potassium is as much important as sodium. Sodium has a little more of a role in water regulation than potassium does. Potassium is massive in neuro and muscular signaling and contraction in muscles. Now we have calcium down here. Calcium is kept at lower concentrations than these others, but it is kept at exceedingly low concentrations inside cells because it is a key, what we call cofactor for enzymatic function. So we keep it in higher levels outside the cell than inside the cell. And it's involved in all sorts of enzymatic functions, like, for instance, blood clotting. It's also involved in release of neurotransmitter from neurons and cardiac contraction and muscle contraction. It's what in cardiac and skeletal muscle, it's what binds troponin and moves tropomyosin to initiate contraction. So it has this important signaling enzymatic role in cells and tissues. We store a lot of it certainly in the bone, hydroxyapatite type storage. And it's controlled by a hormone called parathyroid hormone, or we just abbreviate that PTH. As I said in the previous slide, sodium is a big factor in regulating water fluid from compartment to a compartment. And this is why when we want to fluid challenge a patient, most of the time we're going to give a 0.9% saline solution, which is roughly 300 milliosmolar. All right, so if we need to add volume to a patient, like for instance, if they've had blood loss or they're dehydrated, this is what we call an isoosmotic solution. It is roughly the approximate osmolality of normal tissues. You can also do it with 5% glucose. That's much less common, but it can be used as well. Why the percent differences? Because of the molecular weights of the compounds, and when you calculate osmolarity and osmolality, you calculate based on molecular weight. All right. If we change the solute concentration one way or another, either because of dehydration or too much water or therapies and so forth, we get net water flow. And that net water flow will move into the compartment that has more solute. So if you have more solute, you have less water. So osmosis is the movement of water down its concentration. So if you gave somebody pure water, one of the things you could expect is cellular and tissue edema, because that pure water is going to diffuse into those compartments. And people have actually died drinking too much water from literally water toxicity. So we always try to keep the compartments isoosmotic, or it's isosmotic here. These are 2 examples of those solutions which are considered isoosmotic. If we have any deviation from the isosmotic paradigm or isosmotic situation, we get changes in the cell volume, the tissue and cell volume. So if we are too concentrated in solute, like for instance dehydration or blood loss, what will happen is water will leave the cell, and that will lead to a term called crenation. If you look at a red blood cell, look up crenation. The red blood cell will shrivel like a raisin in the sun. All right, a raisin is nothing more than a dehydrated grape. The water has left the tissue of the grape and now it has shriveled. And that's what crenation is. And that can happen not only in blood cells, it can happen in other tissues, and you have loss of function and organ failure. Or if we have the other side, if we have too much water, not enough osmolarity, same story, the water will diffuse down its concentration into the cells in tissues, causing swelling, which can, if left unchecked, can actually rupture cells. If you add pure water to a smear of red blood cells, they will swell and rupture in very short order. Here is one of those slides we want to be very, very careful with, and I think the key here is to, again, re-paraphrase this. I think this is written a little too technically. If we need to retain volume, if that is to say we need to maintain or increase blood pressure, that's normally considered a sympathetic response. And what happens is that sympathetic response leads to release of aldosterone and anti-natriuretic peptide. These are water conservation hormones, all right? And we talked about aldosterone earlier. So, and now we have this other hormone like peptide. It's a sympathetic response that leads to release of aldosterone and retention of sodium, which means retention of water. Okay, that's the simple message. We have a sympathetic. Let's not worry about all the in-betweens here. That leads to retention of sodium through aldosterone and anti-natriuretic. Anti-natriuretic means against sodium in the urine peptide. And here's another one that, again, could go deeper into the rabbit hole than I think we really need to go. But it is important to understand what the thirst response is. One thing you might notice if you watch any movies or that have people that have been shot but haven't been killed outright. One of the things you will note about people who are losing blood in particular, they will complain of extraordinary thirst. They are extremely thirsty because the thirst response is one of the ways that we trigger behavioral patterns in order to acquire water, which is what we need in order to mitigate the dehydration response. So if we have low blood pressure and or high osmolarity, the hypothalamus will then send out signals in order to try to A, reduce water loss, and B, trigger behaviors and desires in the higher order neurons to acquire water. And that's why somebody who is losing blood will complain of desperate levels of thirst. Okay. So in dehydration, what happens is we get too many solutes that will affect protein function and compromise the ability of the organs to go. That's what we call a hypertonic situation. The cells shrink, that's crenation. So what we have increased osmolarity, that leads to, and decreased volume, that will lead to the water-seeking behaviors. They'll feel tired, sleepy. Sometimes you'll feel hungry too. People sometimes misinterpret the sensation of thirst for hunger. It's kind of weird. There certainly you'll have reduced urine. That's a slower response. All right, it'll be darker. I actually was caving in New Mexico in a cave. We were down underground for days, and I got pretty dehydrated. And it was a pretty profound change in the urine consistency and color. Certainly will feel headache. You might be dizzy. You have a lot more vertigo when you move around. And again, severe dehydration will lead to compromise of protein function, which then could lead to organ failure and death. Overhydration, what we have is we have too much water, not enough electrolytes. So we are hypotonic and the cells are vulnerable to swelling. And again, we can see some similar symptoms because we're compromising function. Maybe we have some neurologic symptoms. Okay, maybe confusion as opposed to headache, loss of consciousness, maybe vomiting. That's one way to get rid of volume. And again, you can have neuromuscular issues that lead to seizures, coma, and death. So just like pH or just like blood sugar, blood oxygen, water is one of those things, maybe a slightly slower than some of those other ones, but can lead to very extreme situations. So focus on this on the left. We're not too worried about all of the processes over here on the right. So in the previous slide, we focused on the consequences of loss of water regulation. Well, one of the key ions we have yet to talk about is the proton, okay, which is what gives rise to the abbreviation pH right here, pH. This is the concentration of protons. That's written in a type of shorthand. I would love to get into the mathematics of this, but that's irrelevant for us. But there is a little bit of a counterintuitive thing because of that shorthand. A higher pH means a lower concentration of protons. We go from basically 1, you could go to 0, that's very difficult to achieve, and certainly physiologically that's difficult to achieve. The scale goes from 0 to 14. And so a higher number is a lower concentration of protons. All right. A lower number is a higher concentration of proteins. If you get a really concentrated hydrochloric acid, it looks like maple syrup, pH of 1 like that. That's because literally it's just screaming full of protons. All right, very relatively little water. But for our purposes, for normal physiologic function, with the exception of like stomach acid, your cerebrospinal fluid's a little more acidic than your extracellular fluid is. There's some deviations. It's really good to have just a basic handle on pH. And our magic number is somewhere around 7.4. This happens to be an important number for when bicarbonate either gains or loses a proton. And that's sort of what establishes this 7.5. We can get as low under normal circumstances as 7.35. We can go as high as 7.45. We very rarely, especially when we're talking about extracellular compartments, deviate from that. Urine can be a little more acidic. Blood, cerebrospinal fluid, pericardial fluid can be a little more acidic, but not a ton. So here's we can see around pH of 7 in intracellular fluid. So inside the cells, it's a little bit more acidic, but not a lot, just a little bit. Okay. Every whole number, one more thing to remember here, every whole number is a tenfold shift. So for instance, if we go from pH of 7 to pH of 6, we now have tenfold more protons than we had at pH of 7. And if we went to pH of 5, that's 10 times 10. That's 100-fold. This is like the Richter scale in earthquakes. An earthquake with a Richter scale of 4 is 10 times stronger than a Richter scale of 3. It's a logarithmic progression. If we get outside of 7.45 in the blood plasma, technically a little sooner than that, we call that alkalosis. Right, so if we have a reduction in protons, a higher pH, alkalosis. If we have an excess of protons, a lower pH, around 7.35 or so, both of these numbers are getting kind of scary if you ask me. That means that we are acidotic. That's where those terms come from. We will die if we get much far beyond that. We get below 6.8 or above 8.0, that's pretty much a recipe for death, because proteins and enzymes are intimately dependent on appropriate pH to have the right structure. If they don't have the right structure, they don't function. And if we don't function, we die. So how do we regulate acid-base balance? Now, there are multiple mechanisms, and it is an overlapping and redundant system. We're going to look at some of the chemical ways that we can regulate acid base in the body. There's one big one and two also important but much smaller ones. All right, so 3 major ways that we can maintain chemical acid-base balance. Another way that we can maintain it is through respiratory mechanisms, but right now we're going to focus on kidney and what we call metabolic mechanisms. The chemical ones, the big kahuna of them all, is bicarbonate. Rapid first line of defense happens literally in split seconds. Bicarbonate is the big gorilla of the 3. Phosphate in intracellular fluid is also the next big one. And for both the extracellular and intracellular, proteins can play a somewhat minor role. So it's bicarbonate in the extracellular fluid. That's the big giant one, the biggest one of all. Then we have phosphate in the intracellular fluid and a little bit of contribution from protein in both intra and extracellular compartments. Your brainstem, and by a certain degree the hypothalamus, can sense pH quite well. They're very, very sensitive. So these ones here are waiting to happen. They're already in the extracellular and intracellular. They don't have to be released. They just behave in the way that they chemically behave. But your body's also going to take some proactive responses as well. And if we start to see, if the brainstem starts to see a change in pH, it will send responses to muscles and tissues in order to help the chemical buffer systems do what they do. The slowest one is the kidney. It is super duper powerful, but requires several hours. That's because you're going to be using hormones, which require changes in genetic expression and moving of transporters and things. So oftentimes it takes a minimum of hours and often takes days. But it is the most powerful and it's the most long-term of the 3. Okay, everyone, we've been going for a little more than half an hour, so I think it's time for a little break. I'm going to pause right here and I will pick up where we are leaving off here in the next video installment. So take a quick break, think about what we have talked about. Before you just bust out of a lecture, you should always just take a minute or two to sort of reflect rather than going straight to your phones. You guys, if you go straight to your phones, you will literally are just ejecting some of the things that you just learned a few minutes earlier right out of your brain. So take a quick break and I will see you on the next installment.